when you take cell wall, you have the definite shape and protection will be given to cell, especially plant cell by cell wall. Then the cell wall forms the backbone of the plant cell and providing tensile strength and mechanical support to the cell. Okay. Then the movement of different materials like solute molecules, solvent molecules, all these things happen through cell to the cell from environment. Then we discussed about the apoplast word. If you remembered, I used a term called plasmodesmator. You remember the word plasmodesmator?
Yes, ma'am.
So plasmodesmator, it acts as a cementing substance. Like it helps in connection between various cells that were far away from each other. So this plasmodesmator functioning is related to the cell wall nature only. So that word stands for by the cell wall. Okay. Then this is the point which I left because of a problem with uploading the slide in the last class. Okay. So here, if you see, a word called turgor pressure is there. Okay. Now, let me explain first this word. Then we'll get into what does mean by contracting this turgor pressure. So here, if you take a cell, suppose assume that this is a plant cell, and in plant cell you will have a big vacuole, isn't it? So this might be the cell wall. Assume that this is a plant cell. Suppose if the outside environment of the cell have high concentration when compared to inside environment of the cell, then what happens is from outside you will get flow of, presuming that this as a cell with the vacuole inside, and when outside environment concentration is more than the concentration of the cell inside it, what happens is there will be flow of the materials from outside the cell into the cell. So as a result, what happens is the cell vacuole will get increased in its size. This condition of cell we call as turgid. Okay. So the turgidity of cell happens when the extracellular material which is outside the cell moves into the cell. So all the inflow of the external material will be absorbed by the vacuole and the vacuole size increases. It goes on increasing. So this condition of cell we call as turgidity. In such turgid cell, what happens is there is a possibility of lysis of cell. Now, lysis means what? Rupture of cell, because the vacuole size getting increased and it pushes the cell wall. As a result, the cell may get disintegrated or ruptured and ultimately result in death of cell called lysis of cell. So how a plant can avoid such conditions? See, when outside environment changes due to high temperature or high humidity or heavy rainfall, in all such conditions, turgidity of cell may occur. So if turgidity of cell happens, the vacuole size increases and the plant cell may go for lysis and ultimately death of the cell. So this can be avoided because of a pressure called turgor pressure. If we assume the outer one as cell wall, every cell will have cell membrane inner to cell wall. This is what In the last class I explained. Okay. So if you assume that this inner one is the plasma membrane, the outer one is the cell wall, when the vacuole size gets increased, the plasma membrane applies some pressure on the cell wall, like this. If you assume this as plasma membrane, it applies some pressure on the cell wall. Okay. So because of that, the turgidity of the cell can be balanced. This is how a plant cell manages when the environmental conditions changes. See, you got the point? The plasma membrane will apply some pressure on the cell wall in order to balance the incoming pressure. Okay. So here, this word, the turgor pressure is nothing but turgor pressure is the pressure applied by the plasma membrane on the cell wall. Okay. Because of which the cell will remain intact without having any disruption or lysis when environmental factor changes. Am I clear with this point?
Yes. Yeah.
Any doubts with the counteracting turgor pressure word? Because this will again see in plasma membrane point also. Excellent doubt. I was expecting this only. Very good, Kavya. Yeah. See, when the vacuole size gets increased, the contents in the vacuole has to be released, isn't it? In order to maintain its normal size. So when plasma membrane is applying pressure on the cell wall, that gives some kind of balancing capacity to the vacuole to release its fluids outside slowly so that it will maintain its normal size. If there is no turgor pressure by the plasma, the vacuole goes on increasing its volume. It won't get a chance to balance the contents inside it. So the vacuole has to get a chance to balance the content by releasing the accumulated outside. Yes? Got it, Kavya? Arya, you typed
Mama, I didn't got it.
Not that you didn't got it. See, what we are talking that the vacuole size gets increased because of the outer materials are coming inside. Okay. Now, these materials has to move outside. For that purpose, vacuole has to get a chance in balancing the pressure inside the cell. Isn't it? So, the balancing of pressure inside the cell will not be done by the vacuole itself. It has to be done by some other part of the cell, which will be done by the plasma membrane. Okay. So, when the plasma membrane pushes the pressure volume outside the cell, then the vacuole will get a chance to adjust the contents inside and slowly releases its contents to outside of the cell. Okay. Then the cell will get into the normal size of the vacuole. Abhijit, still you didn't get?
No, actually, I'm a new student. I did not understand what.
Okay. So, Abhijit, here we discussed about the plant cell structure. We started it in the last session. The plant, mute yourself, Abhijit. Please mute yourself. Okay, thank you. So, the cell wall, how the cell wall functions in protecting the plant cell from outside environment, that part we are discussing from the last session. Okay. I think the recordings of the class are available. You check with the management of UPrep. Okay.
Ma'am,
Yes, Ronak.
Ma'am, what are lysis?
Lysis is disruption of the cell, or in other words, you can say death of the cell. Lysis, breaking up.
Okay, ma'am.
We are done with cell wall. Are there any doubts with cell wall still? If you have any doubts with cell wall, let me know, because we are moving into the